This
I
conference
would
will now be recorded.
I would like to welcome you all to this very special session on the signal detection, signal evaluation, and management. This is our eighth webinar in the last about 2 months, which we have focused on several issues within the clinical trials and pharmacovigilance domain. Every adversity, presents the opportunity, and uh, due to the COVID 19, the way we have started working, the way we have started interacting with our community, our friends, our colleagues in the industry, it has changed completely. And in the months to come, I think this process of interacting using the technology is only going to gain further. So it's an adversity which we all are facing. And I wish you all the very best. Be safe, maintain social distancing, and keep your family safe. Today's topic is a very important topic on the signal evaluation and management. As you are familiar, all of you are from the pharmacovigilance domain. Signal detection and assessment is a very important aspect of pharmacovigilance, which plays a very key role in ensuring that the patients continue to receive safe drugs. And there is no static sort of a drug safety is not static that you do the during the clinical trials and you will continue to the drug continue to be safe. No, with the changing times and the volume of patients, changing geographies, changing practice habits of the medical community brings in the lot of new signals, and that's where we have invited an eminent speaker, our expert on pharmacovigilance, senior drug safety physician, Dr. Anu Dambalkar, who will Anu will speak about discuss about the signal evaluation and management. This is the basic sort of an introductory session on the signal evaluation management. And at the end of the workshop, this would be made available on our YouTube channel at the Cliniminds. And in case anyone wants to refer this later, it would be available. Uh, During the program, we would also share with you a safety database which has got the signal management tool available. And my colleague today, Mr. Kuldeep Kedja, who is our senior pharmacovigilance manager here at Cliniminds, would also take you through that safety data base with the feature of signal management and evaluation. So I would like to now hand over the session to Dr. Anupama to take us through her session today and contribute to this, knowledge, spreading this knowledge further. Thank you, Dr. Anupama. Please, I am handing over to you now.
Thank you, Kamal. This is Dr. Dambalkar. Uh, so I hope you can see my screen. So let us just start with the topic. Today we'll be talking about signal detection and management overview, just a basic overview of the topic. So what is signal detection? Why it is important in PV? Since you all are from PV background, so you must be having some basic idea that signal detection is important because it involves looking at the adverse reaction data for patterns which suggest a new safety information. So as we all know that safety information for a drug is never constant. It's a rapidly evolving field and every time we can get a new safety profile for a drug. So signal monitoring, signal detection and its evaluation is very important. Mostly it is associated with the drugs during the post-marketing phase, but it's not limited to, and it can be used during the pre-marketing clinical trials also. The definition of signal, it is much debatable and we have multiple definitions, but I personally stick to this one, which is provided by the SIOMS working group 8, because it actually provides you everything what a signal is, as it says that it can arise from one or multiple sources and it can suggest a new potential causal association or a new aspect of a known association between an event and adverse or beneficial, Just to highlight this point here, I would request everyone to put their mic on mute, please. Uh, Thank you. So just to highlight this point that if you are getting any beneficial event from a product which was not labeled or which was not expected, even that initially we have to consider it as an AE. We do consider it as an AE, and that too will be associated with signal detection. So that is finally to be judged of sufficient likelihood to justify a verificatory action. This is a definition of what a signal is. There are various other signals definition from WHO, from EMA. So this is just for reference. And as different as these definitions might appear, but one common thing is that what is a signal? That it is information from different sources about a potential harm that may be caused by a medicine. So to conclude, signal is a work hypothesis. So basically, we are just generating a hypothesis that yes, this could be a signal. Further, it is a signal or it is actually a safety profile needs to be assessed. So you have to further work on it and then analyze it and then finally decide whether it was actually a safety issue or not. So what these are the basic type of reactions which we see for a drug. Like type A is augmented, which is predicted from the pharmacology of the drug. Type B is bizarre, like for example, anaphylaxis, even a small dose resulting into an event. We classify it as bizarre. Then there is chemical or chronic, which is related to metabolism, like if hepatotoxicity is caused by the drug because it is metabolized through liver. So that reactions we call it chemical or chronic. Then type D is delayed. The best example for a delayed is after a long-term treatment of a drug, we sometimes encounter cancer, like with phosphomide. Cyclophosphamide causes bladder carcinoma. So this is a delayed or after long-term treatment. Then type E is after end of treatment. This is a typical example of drug withdrawal, in which the reaction occurs after the drug is withdrawn. So, what are the examples of safety signals? So, of course, the best and mostly encountered example is something new, serious, unexpected adverse event. Something which is unknown about a drug safety profile is the most encountered safety signal you will see. Then, even if it is known, but there is an increase in frequency. Suppose if a drug was reported headache as an uncommon event, only one
Ma'am,
in
can you
100.
kindly present in full screen mode? Thank you. In the full screen mode.
You can press F2 for full screen. Yeah, you can do the slideshow for.
Click on present from current slides. Carry on, Dr. Anupam.
the occurrence of adverse events increased, that will be safety signal too. Any new drug interaction or device interaction, any drug or food reactions, all would count as a safety signal. Then identification of any previously unrecognized population. As we all know, there is some population, for example, pediatric group most commonly is in most of the drugs, it is not an indicated population. We have special set of drugs which are indicated for children. If you get something which is in the children population, in the pediatric population, or in any population in which the drug was not recommended or not given before, and you get some clusters of report from that population, that would count as a safety signal. Any confusion about product's name, this will, I know, if people around you must be knowing that it mainly comes in the product quality issue, but still, if a product quality issue is causing confusion or package labeling which could result into a safety issue, that again counts as a safety signal. Then concerns arising from the way a product is used, for example, medication error, off-label use. Medication error could be any change in dosage, any change in route of administration, which is not recommended, and that can cause a safety issue. That would again cause a safety signal. Off-label use again could be the same. Any change in dose, any change in the mode of administration, any change in the risk population, that would be called as a safety signal. This entire list is not limited, and there could be any other concerns also identified by sponsor or regulatory authority. In signal detection, but how do we detect a signal? So signal detection can be from many sources. There can be cluster of cases from a particular area, or there could be a literature report, or there can be multiple sources of signal detection. For example, mainly, sorry, mainly what we see signal detection usually comes from ICSRs. Um, Please put your mic on mute. Thank you. So mainly signal detection are retrieved from the ICSRs. Then the statistical methods are used. We will talk about the statistical methods later in this session. Statistical methods are used to see whether the events reported are actually representing a disproportionality. By disproportionality, we mean that if suppose we are talking about the example just we gave of headache, if we are saying in a general population who is not taking any drug, headache reported was 10 out of 100, and in the population which was taking the drug, the event reported was 10 out of 100, then there is no disproportionality between the population which was taking the drug and which was not taking the drug. And if it was 20 in population taking the drug and 10, then there is a disproportionality. It is increased. So this is a common example, like a vague hypothetical example I'm doing, but this is how statistical methods calculate a rate. And based on that, they assess that whether it is a signal or not. So these are the two basic methods, the qualitative and the quantitative method of assessing or detecting a signal. Then this is just the details that how we set a threshold for statistical measure, and like I just explained, and then we decide based on that. If suppose the analysis at the end based on the statistical, the qualitative and quantitative is inconclusive, if you are not getting, then maybe you require additional data. You can go for data mining for more cases. You can go for literature search and search if you have any relevant literature. You have multiple other datas like WHO Vigibase, the FAIRS data, and you can just go in your analysis further to see if it is inconclusive in starting these two process. Then this is just the sources of ICSR, because ICSRs are the main sources for detection. So these are like unsolicited, solicited, contractual, and by regulatory authority. These are the main sources of ICSR. So as we said, signal detection should follow a methodology which takes into account the nature of data and the characteristic, like time on market. If suppose we are saying the drug was in market for last 10 years, and the signal is coming now, and if we say that drug was just for 1 year, so these two things will make a lot of difference when you are going for the data. The time the drug is in the market will make a lot of difference in the number of cases and number of literatures and all. So the larger the data is, it is easier for us to detect a signal because the statistical data, they give a more accurate result. So patient exposure, again the same. The larger number the patient is exposed, we have a better results as when statistical data is concerned. So as well as the type of medicinal product concerned. So of course the vaccines and the biologicals, they for example may require specific methodological strategies because they have a stratified population. Vaccines are given to certain set of people. It is not something which is commonly. Again, biological medicinal products would be mainly given to cancer patients or the other immunological patients. So this set of patients in themselves, they have certain conditions. So the existing condition of a patient would always confound for the given factor. So in that you have to have a special methodology for assessing. Then signal detection may involve, this is the just review of what we all discussed right now, that it involves review of ICSRs, the statistical analysis, combination of both depending on the size of dataset. Okay. So these are the common statistical signal detection methods which we were talking about, like proportional reporting ratio, proportional odds ratio. Usually greater than 1 is considered significant. So for this statistical methods, we have our software in Cliniminds for signal detection, which we will be taking at the end of this session. Mr. Kuldeep, he will be taking you through the software. So this is a brief about how we have discussed about it. So it is a brief that how statistical signal detection system works. So it is basically a calculation which you don't have to calculate because it is already in the software. So we just at the end we put our values there and then we get if it is significant, the ratio is obtained. So if we are getting something significant from the statistical signal detection method, then you actually further analyze it. Statistical cannot be the solo criteria for detecting a signal. It is always assisting you for your further analysis. It will just give you the initial detection part. So it always has to be, when you come to the analysis part, it has to be combination of the manual clinical method and the statistical data. So if you can just see, these are the tables, these are the formula which is generally used to calculate. So this is all feeded in the software and you just have to put the value and then you get the result. So this, in signal detection method, we are just taking an example like we discussed just now, that suppose in the product taking people, some incident, say incidents X, a adverse event X is common, and the background incidents of the event in the people who are not taking is rare. For example, it is phocomelia due to thalidomide. Phocomelia is rare in people who are not taking thalidomide, and it is very common in people taking thalidomide. So an ease of proving an association becomes very easy because you know it is not common in the background. And signal detection method here would be normal ICSR or periodic review is enough. Second scenario, in both the cases, people taking the medicine and not taking the medicine, in both cases it is rare. Then it becomes little less easier. You have to go into details of everything and see that whether it actually is precipitating the event, the drug taking. For example, race syndrome and aspirin. So in this also ICSR or periodic review would help, but it is not as easy as the first one. Then third would be common. If both the events are common, cough for example with AC inhibitors, it is common in normal population also. Cough is a very common thing and it is common in people taking AC inhibitors. So in these cases, it becomes very difficult, and in this, the statistical analysis is very important. So you go for a large observational trials or large data, and we do data mining, which is gives you a very relevant result. Same is with if it is uncommon, if the scenario gets opposite, that it is uncommon in people taking medicines and common in normal population. It is something very rare, but it does happen, like for example, breast carcinoma and HRTs. So in this, it becomes very difficult to assess, and this again, you go for large clinical trials and large data and data mining for detection. The last scenario with rare and common, it is nothing, no example is known yet, and I think it is virtually impossible to detect or proving an association. If you want to get further idea about the causal association and stuff, since it's a small session, we cannot go into a very detail. You can further read this. We have a reference here. So signal prioritization. Suppose if you have a single drug for which you have multiple signals. It's not a very common thing, but it can occur that you have 3 or 4 signals from a single drug, and you have to prioritize, because signal is something which is you have a particular timelines and you have to finish within that timeline. So how should you prioritize when you are evaluating? So you have to evaluate the impact of the signal. What is the severity, the seriousness? How would it affect the patients if it is not assessed? This is the first criteria of evaluating the and prioritizing the signal. What is the patient exposure and the estimated frequency of the adverse reaction? How will it affect? How many people will it affect? Then the patient exposure in vulnerable population. Suppose if you have a pediatric group being affected, you do prioritize it before all other signals because it's a vulnerable population. Then the consequences of treatment discontinuation on the disease under treatment and availability of other therapeutic options. Suppose if it is a drug, it's a painkiller. You have other therapeutic options. It is not something very important, but you have a cancer drug for which there is only one drug and you don't have any other option. Then it becomes the priority. Then expected extent of intervention, like how can you actually minimize if it is a signal and if it is impacting the how actually you have the methods of minimizing that risk. So if you have it, that expected extent, how can you do it? Based on that we prioritize. Then whether the signal is likely to apply to other substances. Suppose if it is a class effect, if it is macrolides which is happening, so every antibiotic in macrolide groups would be affected. So you prioritize so that you can modify for every drug of that particular class. And this is something for current scenario, that in some circumstances signals it could cause much media attention or public concern. So the MAH, they take a appropriate time frame so that they prioritize and do it initially. So we are done with the signal detection. So after detection, what do we do? This is the steps followed after signal detection, that a signal is validated. If a signal is validated, it is further assessed considering all the available data. If it is not validated, then the cycle stops here. After assessing if the signal is validating and after further assessment, do we consider it as a risk? If no, then the signal is being refuted. If yes, then we propose actions such as labeling product change or issuing a letter to the healthcare professionals or other risk minimization measures as important. So after validation, let us move to if the signal is validated, then we evaluate the signal. This is the most important part of a signal. So once they are identify, you have to evaluate whether it was actually a signal or you have to refute it. So, for evaluation, we take all the data possible. The safety data would involve the data mining, the ICSRs, the main source, the literature in available, the literature search is done, and the literature is reviewed for any potential causal association. Then even for the clinical trials and toxicology, this is not applicable for all drugs, but this can be applicable. So all the data available to you, we actually do a manual ICSR review, a statistical review, and a combination of both for further evaluation. This is a criteria for causal association. This will apply to any event and drug relation. That what we actually see when we are evaluating a case, then what we actually see for causal association. So it is a universal, the Bradford Hill criteria, which we see, which is the strength of association, the consistency of association. So the strength is like how strongly you can say that a drug was given and the patient developed that event, and the other patient who was not taking the drug, nothing developed. So it could be strongly associated. The consistency, that every time he is taking, every time he is developing it, it is like very consistent. Then specificity, it is being developed with only that drug and not with the other drugs. Temporal association is very short latency. You're taking the drug and it is being developed in like 30 minutes, 2 hours, depending on the pharmacokinetics of the drug. It is developing. Then biological gradient is depending on the biological of the drug, like what is the mechanism based on that. The plausibility is again that it is plausibly associated, it's commonly. Then it should be coherent, and the reversibility is that the when you stop a drug, it gets reversed. There is experimental evidence to it that maybe in the toxicology study, the animal studies, you can actually recreate the result. So these are the set of uh, association criteria which we actually use to assess causal association of the drug and an event. Then classification of signals by strength. After you have assessed the signal, you have evaluated, these are how we classify the signals. Like what are strong signals? The cases with no alternative causes, a fairly strong that you can have alternative causes, but you cannot actually just refute that no, it was not because of our drug. So one important criteria in this is a positive D-challenge. I think everybody must be aware of what a D-challenge is, but still, in brief, that D-challenge is that when you just remove the drug, then the event stops. A re-challenge is when you reintroduce the drug, the event again restarts. An average strength would be that you have both the things. You have factors, risk factors, as well as you cannot completely refute the drug thing. Fairly weak is that the all other risk factors, they are more strong in causing the event rather than the drug of interest. A weak is that it is either you have insufficient information to assess, or it does not, it lacks the criteria. We just saw the Bradford criteria, it lacks in the parameters from that criteria, and you can explain it by alternative causes. Then finally, why are we doing all this? Why are we assessing? What do we do after a signal is evaluated and you have confirmed that yes, it is a safety issue, it is a signal? So we set up a study specifically investigating a particular safety concern. That is, study specifically means, suppose you have now seen that yes, it is, so you further monitor it. You develop a strategy which further monitors that particular drug with that particular event. Then you review the benefit and risk of the product, and based on that, a further modification is needed. Whether you have to suspend or revoke the marketing authorization. Most one of the most commonly which you will see after signal is to propose a change in the labeling information. This is something which is most commonly encountered in practical scenario. Or to issue a warning to the healthcare professions in form of a DHPC, the Dear Healthcare Professional Communication. Or perhaps if you think it is not a very big issue, but still it has to be reviewed, then it is constantly monitored in the coming aggregate reports like PBRAR. This is the recommendation on signals from the Pharmacovigilance Risk Assessment Committee, that a MH, and this list is not inclusive of all. There could be something new always from the health authority, but this is a general list what is recommended. That the MH should provide additional data for assessment. It should just provide a review of additional data on the following PBRAR, or submit if they want the information just within the time frame, which could be either it could be 60 days, it could be 90 days. Then you prepare a separate report for that particular signal. It could be called anything. An ad hoc report means that, and an ad hoc report is prepared for that particular signal and submitted to the authority based on the timelines provided. So here is the role of us, the PV people, that finally why are we doing it. So these are the recommendations, and these reports we prepare and submit it to the health authority. The MH has to update the product information. So this is again, a report is prepared, the labeling working group is being provided with the report, and the labeling working group would update the label. The RMP, the MH should request to if there is no RMP, then you have to prepare a RMP. This is RMP is a risk management plan, or an existing RMP has to be updated based on the new assessed signal. Then the MH should implement additional risk minimization measures if necessary, like a DHPC or educational materials for healthcare professionals. Then it can also, the MH can also the pass protocol study, which is a post authorization safety study, and submit the results of that study to the authority. Then it can collect further information. The other EMA expert group should be consulted. Even an inspection could take place to verify that MAH satisfies the PV requirements, and any other appropriate action that is not listed. Sometimes even no action is required if you think that, okay, the safety profile of the drug is not being hampered. Sometimes it is not required, no action from PV. You just submit the report and the cycle ends there. So this is a brief idea about what is signal detection, how do we manage it, and finally, what do we do it after management. For the software, which we have a tool for signal detection, Kuldeep, our senior PV manager, would be taking through. I'm just giving him the presenter rights.
Dr. Anupama,
Yes.
this is Dr. Dundesh here.
Mm. Yes.
I just had one question actually. How do you decide that PRR should be taken more than one or more than two? Proportional reporting ratio or reporting odds ratio? How do you decide it actually?
PRR generally considered greater than 1 is considered significant, and greater than 2 is considered for the signal detection. It is considered confirmatory for an increased rate. So it is
Okay. Is there any
yeah.
is there any guidance given? I just wanted to understand. How do we consider more than one or more than two?
See, you for the statistical analysis, of course, you have a separate set of guidance, and one thing is also there that it actually varies from company to company. So even the reports
Okay.
being
So
prepared,
because
even yeah, so it will vary from MH to MH.
no, that's what when I encountered a scenario where we have considered more than two actually. PRR more than
Yeah,
two.
yeah, that I told you that actually greater than 1. See, if we are considering it for detection per se, and then it is considered greater than 2. But greater than 1 will always give you a, you can say, a warning sign, like it is considered significant. But when we talk about
Okay.
proper signal detection, when you have all the data, and when it is
Okay.
statistical method is done, then greater than 2 is taken as a detection part.
Okay, so it's applicable
Hi.
to only PRR or ROR also. ROR also actually.
Dr. Dundesh, we need to move forward. We will take perhaps offline after the session. We will take this with Dr. Anupama offline. Yeah, so that we can
Okay,
move on
okay,
right
Kamal.
now with
Thank you.
the session. Thank you, Dr. Dundesh. Thank you.
No problem. Thank you. Thank you.
Hello everyone, good afternoon, this is Kuldeep, and thank you Dr. Anupama for explaining about signal detection and management. So here is the software safety database that we have. This safety database name is Safety Easy, and it is by AB Cube company. It is a French company that we have, and this software have the functionality of from beginning, like from ICSR, case processing, and then medical assessment, aggregate reporting, report generation, line listing, and also have the functionality of signal detection inbuilt. So for as informed by Dr. Anupama, also we have different softwares for it, like we have Argus for data entry purpose for the case processing, and for Oracle have a different software for signal detection. This signal detection is not inbuilt in Argus, and that software is known as Empirica. And for RSG, RS Global also have AG signals. But here in Safety Easy, we have this signal detection software that is inbuilt in it. So let me take you through it. You as you all are PV experienced people, so you all know what is ICSR case processing and all. So I will directly go to the signal detection. So first, we have this functionality here. From this report generation, there are different functions that are there in this software. From here, we can generate different reports of like PSUR, DSUR, and February and all. And here only we have the functionality to calculate the PRR and ROR both. So this software basically gives you the PRR, or if you want the ROR at the different occasions. So here I have generated a report, already generated a report for PRR, proportional reporting ratio. Here this report looks like, here are the filters which I have put in. This is the calculation part that they have taken in background. So here we have, so this PRR, we have not put any filter in it right now, any product filter or any event filter. We have just generated the report of this PRR from last 3 to 4 years. That is data that is present in this software. So it gives you data with respect to the SOC. According to the MEDRA SOC, it gives you data. And the PRR is as per individual PT that are already reported in the software, or in the cases that are already entered in this software. So here you can see product issues, SOC have PT which are already reported in this software. Product physical issue, we have PRR. The PRR is calculated according to that, and we have KI2 also. KI2 is also calculated, and these are some other statistical things that are calculated accordingly. So main thing that we see here is PRR, that is one. If PRR is more than one for any event, then it will be highlighted red in this report, so that it will give you, or it will reflect the signal for that particular event. And also the case numbers are also here, so you can, or the company can go to those cases and do the signal evaluation individually for them. So this is how the report looks like. Here we can put the filter of particular product also, and it will show the product name here, medicinal product name. Or you can put the filter for active substance, or you can put the filter for also the SOC, which SOC you want. So those things you can calculate. So this is the inbuilt feature in this software that we have to calculate the PRR or the ROR, from where you will get the signal. If it is more than one, then it will show the red mark, and it will be considered as a signal. Then the company, or you have to go to individual cases and evaluate those signals as explained by Dr. Vipra in the presentation. Same way, ROR is also calculated in the software that we have. And in our course that we provide, the course that we have is Signal Detection and Risk Management course, certificate program in signal detection and risk management. This is the combined course that we have here in Cliniminds. In this course, we will provide you the access of this software for detecting these signals. The signal detection process, that the software access will be provided to you, and we have signal detection topics that will be covered during this course will be background to signal detection, legalities in the signal detection, then the objectives of signal detection and key components, signal analysis, the data mining. Data mining is a pretty new thing that is there in the pharmacovigilance, which is used nowadays in signal detection purpose. So that will also be trained. You will be trained on that information component, and these software access will also be provided to you so that you can yourself do the hands-on training on this particular software. Then in the risk management, we have the EU risk management plan and the REMS, that is the risk evaluation and mitigation strategy, that is in US. And also we will train you on how to calculate the benefit risk assessment, how to do that, and how to calculate the benefit risk ratio. This is the part of our course, and the duration of the course is 3 months. The mode is online and also e-learning is there, and 3-month courses it. And about other details about fee structure and other details, our marketing team will contact you individually about it. So now you can, if you have any questions, please write down your questions in chat so that we can take those questions individually. So here we have one question that presentation will be there on the YouTube or not? Yes, Dr. Venkatesh, we will be uploading the recorded presentation on YouTube, and the marketing team will share the link with you all individually. Thank you.
We would shortly advise you in a day or two. You will receive the YouTube access for the program, and you will also receive the course information, and the fee details will be provided shortly.
And we have another question from Bindu Gilbert, cost of the course. So as I just said, our marketing team will contact you individually, all of you, to share the details about the course and the cost of the course both. Then we have how aggregate report and signal is linked. Dr. Anupama, request you to please answer this question, how aggregate report and signal is linked.
Hi. So actually, when a signal is confirmed after evaluation, then it is the ad hoc report if you are preparing for submitting it to the authority, if recommended. That too is an aggregate report because you are taking into consideration all the ICSRs received and evaluation. You have all the data mining results like FAIRS data, wiki-based data results. You have the literature reviewed, and then a report is prepared. And if an ad hoc report is not required, then you have a separate section in PBRAR, if you are aware of the PBRAR format, in which the signals ongoing or closed is presented in the PBRAR. So I hope this answers the question, the link between signal and
Next question, Dr. Anupama. What special methodology is what special methodology is applied for special cases like pregnancy? Question by Dr. Sujay Patil.
Okay, so actually, as you see, pregnancy is an issue in which a special methodology can not be applied. The basic methodology followed for any pregnancy related cases is the cases received. We always keep a track of the this can you can say is a basic review study always in most of the cases, because pregnancy is a condition in which any trial is ethically not recommended. So mostly the methodology applied for pregnancy cases would be review studies, review of the cases received, and
Thanks, Dr.
literature sources, of course.
Another question by Preetham. Is there any chances in current process due to new procedures for NISS by FDA?
Sorry, I am not aware of this.
Okay.
What exactly, like, in what
We
process are you, like, which process, in context of which process are we talking? If
okay.
Preetham can elaborate the question, I
Yeah.
actually don't know what process he's talking about.
He's talking about new identified safety signals.
No, that I understood. Like, changes in what process? The signal evaluation process. That would not
Uh,
change. The signal detection and the validation and the evaluation process, which the process is being followed for signal detection and management, it has no impact.
there was a one question by Nivedita. I would like to know some references for manual signal detection.
Okay. So, manual signal detection basically is the medical review done by the medical person. So, this is something very elaborate. So, this course would actually deal with how do we assess a case, how do we actually, if there are a number of cases after assessing, how do we make it an aggregate form of finally evaluating whether that signal was an actual safety issue or not. So, this course will take over all this.
We don't, Dr. Nikit, we would, we use at present the safety easy. We are considering uh, uh, sort of including Empirica. So perhaps we would inform you in a day or two when we send you the course information. At present, we use the built-in tool which is there in the safety easy of the signal evaluation management. So we'll use that, and we are considering Empirica at present. So we'll inform you shortly. Sheikh, your question is how does this course help you to get into the signal without experience? Uh, this is the whole objective, Sheikh, of the program is to give you the skill set actually which you can apply into a day-to-day job. And when you're applying for the job internally into the companies, or if you're applying for the outside job, you will have a sufficient skill set with the help of this program, because the trainers like Dr. Nabama or the other sort of a trainers will train you hands-on on this. For you, Dr. Anupama, question is, should I consider event mentioned in DME list as signal even if it was not received a single time during my period?
Uh, so DME is actually a list of adverse event which is considered rare, serious, and has a high drug attributable risk, like associated with a high risk. But a signal is not what is DME. I mean, I would ask you not to get confused between the two, because a signal is generated, a DME is already what a list is, but a signal is generated. So, no, you should not consider an event. It is something new if it is coming of an existing say, event or a new event altogether. Only then it is considered a signal.
Uh, there's one more question by Ms. Salman. is there any specific requirement that people should be a doctor to enter into signal management domain, or preference will be given equally to the pharmacist, specifically in India? Anupama, do you have a comment on this? I will add to that, but please speak faster.
Yes, sure. So, Salman, there are specific step-by-step approach to a signal. So, when we talk about the statistical data, the data mining part, and so a doctor is required when the assessment part is there. Only during the evaluation part, it is the specific role of a physician. Otherwise, in several steps, we do require non-physicians also.
Yeah, Salman, I think that answers, because there are a lot of processes which are the non-medical in nature will be definitely pharmacist. As a pharmacist, you have advantage, and if you have the knowledge and skill set of the signal management, signal evaluation management, it will be helpful. And there are several non-medical professionals already working in this industry in pharmacovigilance domain and in signal domain specifically. Thank you. You can. Thanks a lot, everyone, for taking out time on weekend, and we look forward to have more interaction in the coming weeks. We'll send you some more information. If you have any specific questions, you can send us on email, and we will ensure that you receive the response. And in the meantime, my business team would also get in touch with you with the program information in more detail, and also inform you
Yes.
about whether we are going to include Empirica as well or not. So thank you a lot. Thank you, everyone. Be
Thank
safe, be
you. Thank
healthy.
you, sir.
Thank you. Thank you, Dr. Anupama. Thank you for the
Thank you,
wonderful
sir.
presentation.
Thank you,
Thank you.
everybody.
Thank you to all. Thank you.
Thank you, sir.
Thank you, everyone.